Today we're going to discuss the circulatory system, specifically blood. The circulatory system is the transportation system of the body. This system consists of two subsystems, the cardiovascular system and the lymphatic system. In the cardiovascular system, the heart pumps blood through a vast system of blood vessels. As it circulates, blood transports nutrients, oxygen, hormones, and waste products. The lymphatic system helps maintain fluid balance and launches immune responses that protect the body against disease. There are several critical functions of the circulatory system. The circulatory system helps maintain homeostasis by transporting needed materials and by maintaining fluid balance and acid-base balance. It protects the body from disease-causing organisms. Blood consists of red blood cells, white blood cells, and cell fragments called platelets, all suspended in a pale yellowish fluid called plasma. The circulatory system also helps regulate the acid-base balance in the body. The normal pH of blood is slightly alkaline at 7.35 to 7.45. pH is a measure of acidity or alkalinity of a solution. So plasma consists of about 92% water, 7% protein, a sprinkling of salts, and many other materials being transported, such as oxygen, carbon dioxide, other dissolved gases, glucose, metabolic wastes, and hormones. Plasma is in a dynamic equilibrium with the interstitial fluid, bathing the cells with intracellular fluid, which is the fluid inside the cells. As blood passes through capillaries, the tiniest blood vessels, substances continuously move into and out of the plasma. Changes in its composition initiate responses by one or more organs of the body to restore homeostasis. Most of the plasma proteins are manufactured in the liver. There are three groups of plasma proteins: fibrinogen, albumins, and globulins. Plasma proteins, especially albumins and globulins, help regulate the distribution of fluid between plasma and interstitial fluid. As blood flows through the capillaries, some of the plasma seeps through the capillary walls and passes into the interstitial fluid. However, large protein molecules have difficulty passing through the capillary walls, so most of them remain in the blood. There they exert an osmotic force that helps pull the plasma back into the blood. This action is important in maintaining an appropriate blood volume. Plasma proteins, along with the hemoglobin and red blood cells, are important acid-base buffers. They help keep the pH of blood within a narrow homeostatic range. Fibrinogen is one of the groups of plasma proteins, and it's involved in the clotting process. There are three types of globulins. We have alpha globulins, which is prothrombin, which is a protein involved in blood clotting, and high-density lipoproteins, HDLs, which transport fat and cholesterols. We have beta globulins, which have to do with low-density lipoprotein. And then we have gamma globulins, which contains antibodies that provide immunity to diseases. When proteins involved in clotting have been removed from the plasma, the remaining liquid is called serum. Next, we have red blood cells. Erythrocytes are the formal name for red blood cells. Hemoglobin is the red pigment, and hemoglobin is used to transport oxygen. Oxyhemoglobin is oxygenated hemoglobin. Red blood cells also transport carbon dioxide. The average circulating lifespan of a red blood cell is about 120 days. Old or damaged red blood cells are destroyed in the liver, spleen, or bone marrow. An erythropoietin is the hormone secreted by the kidneys that stimulates red blood cell production. A mature red blood cell is a tiny, flexible, biconcave disc. An internal elastic framework maintains the disc's shape and permits the cell to bend and twist as it passes through blood vessels even smaller than its own diameter. This flexibility allows red blood cells to squeeze through narrow capillaries to deliver oxygen to cells in the tissue. The biconcave shape of red blood cells provides a high ratio of surface area to volume, allowing efficient diffusion of oxygen and carbon dioxide into and out of the cell. A mature red blood cell lacks a nucleus and most other organelles, leaving more space for hemoglobin. When blood circulates through the brain or some other tissue where cells are low in oxygen, the reverse reaction occurs. The oxygen diffuses out of the capillaries and into the cell. Oxyhemoglobin is bright red. It is responsible for the color of oxygen-rich blood that flows through the arteries. Hemoglobin that is not combined with oxygen is bluish and accounts for the darker appearance of venous blood. In children, red blood cells are produced in the bone marrow of almost all bones. Red bone marrow is a type of connective tissue found in spongy bone. In adults, red bone marrow is found in certain bones, including the vertebrae, sternum, skull, ribs, and long bones. The red bone marrow has immature cells known as stem cells, which multiply, giving rise to blood cells. White blood cells are also known as leukocytes, and they are specialized to protect the body against pathogens. Pathogens are harmful bacteria and other microorganisms that cause disease. White blood cells develop from stem cells in the red bone marrow. In contrast to red blood cells, which function within the blood, many white blood cells leave the circulation and perform their duties in various tissues. They move through the tissues, flowing like amoebas. As they travel through the body, white blood cells destroy bacteria and engulf dead cells and foreign matter. This is called phagocytosis. Phagocytes are cells that are specialized to carry out phagocytosis. There are 5 main types of white blood cells. The first one is monocytes, and they function to differentiate to form macrophages, or large scavenger cells, and dendritic cells, which destroy viruses. The number of monocytes may increase in monocytic leukemia and certain fungal infections. Next, we have lymphocytes. Their function is to produce antibodies and to destroy foreign cells. Atypical lymphocytes present an infectious mono. Too many lymphocytes result from certain viral infections or from lymphocytic leukemia. The function of a neutrophil is phagocytosis. Too many may be the result of a bacterial infection, inflammation, or leukemia. Basophils may play a role in allergic reactions and in preventing inappropriate clotting. They release histamine, which dilates the blood vessels and makes the capillaries more permeable, which is important in the inflammatory response. Finally, eosinophils play a role in allergic reactions. They release compounds important in certain immune responses and destroy parasitic worms. Too many may result from an allergic reaction or a parasitic infection. Next, we have platelets, also known as thrombocytes. Platelets are not really cells. They are tiny fragments of cytoplasm that are pinched off from certain very large cells in the bone marrow. These cell fragments prevent blood loss. There are 30 different chemical substances that are involved in blood clotting. When a blood vessel is cut, it constricts, reducing blood loss. Platelets stick to the rough, cut edges of the blood vessel, forming a platelet plug that seals in the hole in the blood vessel wall. Tiny fibers are produced which reinforce the platelets and form a strong clot. Platelets in injured tissues release substances that activate clotting factors in the blood. A series of reactions takes place, resulting in the formation of an enzyme known as prothrombin activator. Prothrombin activator catalyzes the conversion of prothrombin to its active form thrombin. Calcium ions and clotting factors must be present for prothrombin to be converted to thrombin. Prothrombin is a globulin found in the plasma. It is manufactured in the liver with the help of vitamin K. In the presence of calcium ions, thrombin acts as an enzyme that converts the plasma protein fibrinogen to fibrin. This fibrous protein forms long threads that form the webbing of the clot. Blood cells, platelets, and plasma become trapped in the fibrin threads, which helps strengthen the clot. Within a few minutes after it is formed, the clot begins to contract and squeeze out serum, which is plasma that contains no fibrinogen or clotting factors. As the clot contracts, the ends of the damaged blood vessel are pulled closer together, and the clot itself becomes smaller and harder. A blood transfusion is the transfer of either whole blood, plasma, platelets, or other blood components from healthy donors to a recipient. Blood components can be separated by centrifuging blood. That just means spinning the blood at a high speed so that the denser components settle at the bottom of the tube. Then platelets and other specific components of blood can be transfused. Whole blood or blood components can be stored in blood banks and withdrawn as needed. Before blood is transfused, the blood of the donor and the blood of the recipient must be carefully matched. If blood is not compatible, a transfusion reaction will occur. This is a serious allergic reaction in which antibodies in the recipient's blood attack the foreign red blood cells in the transfused blood, causing them to clump. Red blood cells may rupture, releasing hemoglobin into the plasma, which is a process known as hemolysis. Although several blood groups are known, the most important clinically are ABO and RH group. Blood types are inherited. Each person has either A, B, AB, or type O blood. An antigen is a protein found on red blood cells that differentiate the blood types. The red blood cells of individuals with type A blood have type A antigens. Individuals with type B blood have type B antigens. Those with type AB blood have A and B antigens. Individuals with type O blood have neither type of antigen on their red blood cells. They are referred to as universal donors because they can donate blood to patients with any blood type. Certain antibodies are found in the plasma. Antibodies are specific proteins that recognize and bind to specific antigens. Individuals with type A blood have anti-B antibodies circulating in their blood. Those with type B blood have anti-A antibodies. Persons with AB blood have neither type of antibody, and those with type O blood have both anti-A and anti-B antibodies. As a result, type AB blood is the universal receiver. Each type of antibody recognizes a specific type of antigen. For example, if a patient with type A blood is accidentally given type B blood, the patient's anti-B antibodies will combine with the type B antigens on the surfaces of the donated red blood cells. This causes the donated cells to clump, which results in hemolysis. Blood typing is routinely carried out by mixing a sample of a person's blood with serum containing different types of antibodies to determine whether or not clumping occurs. This just shows the specific ABO blood types. It shows the blood type, the antigen on that blood type's red blood cells, and then the antibodies in the plasma. Next, we're going to talk about the Rh system, which consists of Rh antigens referred to as Rh factor. The most important type of Rh factor is antigen D. Most persons of Western European descent are Rh positive, which means that they have antigen D on the surfaces of their red blood cells, in addition to the antigens of the ABO system appropriate for their blood type. Antigen D does not occur in the blood of Rh negative persons unless they have been exposed to it. However, once antibodies in Rh blood have been produced, they remain in the blood. When an Rh negative woman and an Rh positive man produce an Rh positive baby, the mother may be exposed to antigen D. A baby's blood does not normally mix with the mother's blood during fetal development. However, during the birth process, a few drops of the baby's blood may mix with the mother's blood. This contact stimulates the mother to produce antibodies against Rh positive blood. If the mother carries an Rh positive child in subsequent pregnancies, her antibodies can cross the placenta. The anti-Rh antibodies cause hemolysis of the baby's red blood cells, resulting in hemolytic anemia. The baby's ability to transport oxygen is reduced, and the breakdown products of hemoglobin that are released into circulation can damage organs, including the brain. Rh negative women are given RhoGAM during pregnancy and within 72 hours after delivery. RhoGAM is a preparation of anti-D antibodies. These antibodies rapidly clear the Rh positive fetal red blood cells from the mother's blood. This process minimizes the risk of the mother's white blood cells becoming sensitized and producing memory cells that might threaten a future pregnancy.